The overall goal of this procedure is to detect differentiated interrenal steroidogenic cells in the developing zebrafish through a simple whole mount enzymatic activity assay.
This staining procedure can help answer key questions regarding the molecular and cellular mechanisms of interrenal gland formation and malformation.
The main advantage of this staining assay is that it's rapid, simple, and reliable, making it a powerful tool for genetic or chemical screens in zebrafish.
This procedure can be performed on transgenic fluorescent reporter lines, marking the kidney and the blood vessels, so that multiple tissues in the same embryo can be analyzed.
In combination with vibrotome sectioning, immunohistochemistry, and confocal microscopy, the vascular microenvironment of the interrenal tissue can also be visualized.
After preparing stock solutions for 3-beta-HSD activity staining according to the text protocol, place developing zebrafish embryos in 0.03% PTU in egg water to inhibit pigment formation. Fix dechorionated embryos, or larvae, in 2% or 4% PFA in PBS, with 0.1% tween 20, or PFAT. With PBST, wash the embryos 4 times for 10 minutes each, ensuring that the samples do not dry out between washes. Prior to the staining reactions, freshly prepare parts A and B in separate tubes, then vortex and centrifuge the solutions.
It is essential to prepare parts A and B in separate tubes, adding all the components straight into one single